Eric is Professor of Ecology and Evolutionary Biology at UC Santa Cruz. He is also the Associate Vice Chancellor for Research, and he is also Director of the Fisheries Collaborative Program, the FCP, which supports research, education, and outreach collaborations between our university and the Southwest Fisheries Science Center of the National Oceanic and Atmospheric Administration, NOAA. So you know how in your workplace there's that one person who you know if you ask them for help or ask them for advice or their opinion, you know they've just got it. And they're doing a million things, but they've always got time for you and they just love helping people and solving problems. And it turns out they do all these other amazing things that they never even told you about. Well, that is Eric. And I very grateful to have him as a partner and a colleague. So in addition to all the administrative work and the leadership work he does, he's a busy practicing scientist. His research program looks at connections between evolutionary and ecological processes in freshwater and coastal ecosystems with the goal of informing biodiversity, conservation, and sustainable fisheries management. So the bottom line of all that is Eric loves fish and water and the outdoors, and I think you're going to get a great sense of that tonight. So please welcome Professor Eric Palkovacs.
All right. Thank you so much, Brian, for the introduction. Thank you for the warm welcome. Thanks, everyone, for taking some time tonight to learn about Pacific salmon recovery and our research and restoration efforts. I'm excited to be here and tell you guys a little bit about fish, which I do love. And people say, I always imagine you with a fish. And I don't always have a fish, but people think I always have a fish because I always talk about fish. So this is really sort of my home turf. I really love talking about fish. So you're going to learn a little, hopefully you'll learn a little bit today. So we're going to talk today about transforming Pacific salmon recovery from genes to ecosystems. So first, I think I would be remiss if I didn't just introduce generally the salmon life cycle. So what makes salmon interesting, by and large part, is their complex life cycle. So many people know, but let's just go over it, right? Salmon are born in freshwater. So they hatch in freshwater, largely in freshwater streams. Some species spawn in lakes, but for the purpose of this talk, let's say they hatch in streams generally, and they spend a few months to sometimes a few years growing in freshwater. And then they undergo a physiological transformation called smoltification that transform them from a freshwater fish into an ocean fish. And they make a migration from their freshwater stream out into the ocean. They make this migration because the growth conditions in the ocean are far superior than the growth conditions in freshwater. So they have an opportunity to start to grow really fast once they hit the ocean. There's a lot of food out there, so they feed and they grow and they live for several years in the ocean before they make the reverse migration back to freshwater to spawn. Salmon will spawn one time in their life. So this is called a semelparous life history. So they spawn and then they die, right? So if you go to a salmon stream in Alaska, let's say, during the spawning season, you'll see salmon carcasses littering the banks of the stream. That's totally normal. That's what you should see in a healthy salmon run, because they spawn, they die, they've invested all their bodily resources into reproduction. So they've given everything they have to their offspring to support the growth of the next generation. This is called an anadromous life history, okay? So this migration between freshwater and the ocean is called anadromy. Let's a little bit go through the evolutionary history of the Salmonidae family. So this family arose during let's see if I can get my cursor on the mark, arose back here. So this is sort of the time period right after the Earth experienced a collision with an asteroid that caused the dinosaurs to go extinct. And after that catastrophic event, we had the evolution of the ancestral lineage that gave rise to all our modern salmonids. The Salmonidae family consists of several groups that you may No. So here we have grayling, we have whitefish and ciscos, and down here are the groups that you probably would recognize more as our salmon and trout. So here we have the genus Salmo. So Salmo, these are Atlantic salmon and brown trout. We have Salvelinus, which is a genus of char. So like brook trout are actually brook char, and lake trout are actually lake char. So they're in the genus Salvelinus. And we're going to focus today on the genus Oncorhynchus. So these are our Pacific salmon. So Oncorhynchus means, in Greek, hooked nose, right? And so that's a description of, you'll see as I go to the next few slides, you'll see why they get that descriptor of hooked nose. And I want to draw your attention to the fact that back here in the Miocene, so we've had Salmon, we've had this genus, Oncorhynchus, in California for 15 million years. And swimming in the rivers of California used to be this giant spiked tooth salmon. So these giant spiked tooth salmon of the Miocene were over 8 feet in length and over 400 pounds. And they had these ferocious looking spiked teeth coming out of their upper jaw, and those were sort of a trait that males would use to compete for mates. And you'd think that this kind of a massive fish would probably be a top predator, but in fact, we think that this large fish was actually a planktivore. And so here you can see these are our Pacific salmon of North America here. So we have chum salmon, pink salmon, sockeye, Chinook, coho, and then steelhead. And steelhead is kind of is an interesting case because they're a little bit different from the other Pacific salmon in that they don't die after they spawn. They can spawn multiple times within their life. And so they make the return migration between freshwater and saltwater several times within their life. So they'll spawn, go back to the ocean, maybe come back a second time, spawn, go back to the ocean. This is the sort of historical, this map here shows the historical distribution of the genus Oncorhynchus. So from Japan up around the North Pacific Rim to Alaska, down the Pacific coast, and steelhead historically went as far south as Baja. And you can see these are sort of the spawning forms here, and you can see why they got the name the hooked nose, right? That is the kype that they develop when they're on their spawning migrations. And as my title suggested, one of our goals is Pacific salmon restoration. So we wouldn't need to restore them if they were in great shape. So they're not in great shape. They have been declining. Those declines have occurred across the North American range. So in Washington, here in California, in BC, and in Alaska, we've seen declines in Pacific salmon across all those regions. So they do need our help, and I'm going to talk a little bit today about things we're doing here to try to help salmon populations. Of course, there's a long history of people and salmon interacting with one another. So people have used salmon as a food resource. There is evidence. The earliest evidence for people using salmon as a food resource goes actually back to the last period of the last glacial maximum. The Japanese Jomon culture developed pottery, and there's thinking that in fact this pottery was really developed to cook salmon as a food resource. The earliest evidence for salmon consumption in North America is from Alaska, and so it's from this site on the Tanana River, which is a tributary to the Yukon River. And importantly, that site is quite far from the ocean, right? It is quite far inland. And so this is sort of a common theme, right, in the story of the ecology of salmon, is they're taking all this marine productivity and they're migrating far up these rivers. And they're basically a conduit for all this marine productivity moving far inland in these river systems. And so people living very far from the coast can actually harvest the bounty of the ocean because salmon bring it right to them. And this evidence that people were using this resource goes back a very long time. And then somewhat more recently, we see the spread of salmon consumption across Alaska, and it becomes very widespread inland, and it becomes very obvious that people living along rivers are using salmon as an important food resource. And that, of course, continues to this day. So why are salmon so important ecologically? Well, they're important ecologically because they play what we call a keystone role in the food web. So here is the salmon, if I can get the pointer on there. And this is the marine food web. And this over here in the green, this is the freshwater and terrestrial food web. And you can notice that salmon play this key role because they are the conduit of connectivity between the marine food web and the freshwater food web and the terrestrial food web. So they sort of classically are prey for large marine predators like killer whales. And if you follow the news, you might have seen that killer whales were wearing salmon hats, right? I don't know if anyone saw that news, but and the southern resident population of killer whales is thought to be very specialized on consuming salmon. So they really are dependent on salmon as a resource. When salmon make their spawning migrations up rivers, they then become prey for a bunch of terrestrial predators, like classically brown bears, right? You can think of the classic brown bear, all the brown bears coming from the landscape and feeding at the salmon rivers. But they're also prey for other predators like wolves and eagles, and all this marine nutrients comes up rivers to support these large terrestrial predators. So those connections are not perhaps too surprising, but there are some even more surprising connections. Salmon carcasses, as they pile up on the banks of rivers, as I described before, and as bears take salmon, and they'll eat part of the salmon, but they'll take the carcass up into the forest. So they don't just eat them all right on the riverbank. They'll take some of them up into the forest, and they won't consume the whole thing. And so you'll find salmon carcasses littering the forests of throughout the range of Pacific salmon, and the nitrogen from those salmon carcasses ends up in vegetation, including in the trees of the forest itself. Insects feed on salmon carcasses, and what feeds on insects? Of course, songbirds. So people have found salmon-derived nitrogen even in the tissues of songbirds, and find elevated abundance and diversity of songbirds along salmon rivers compared to rivers that don't have salmon spawning populations. And then maybe even one more step is plant-pollinator interactions. So some of the key pollinators of riparian plants lay their eggs on salmon carcasses, and the larvae develop feeding on the salmon carcasses, and then the adults of these flies will pollinate the plants in the riparian area next to the spawning streams. And so you'll have these flies covering these flowers, and what's really fascinating and amazing about this is a co-evolved relationship such that these flowers bloom at the same time that the salmon run up these rivers, so that it's synchronized, so that when the carcasses are available for the larvae, that's when the flowers bloom, that's when they can get pollinated most effectively, because that's when the pollinators are most present in the environment. So we have this sort of coevolution between run timing of salmon, the life cycle of pollinators, and the flowering timing of riparian plants. So this is a well-tuned coevolutionary relationship. Okay, so the key to what I'm going to talk about today, and really what we're working on today, relates to salmon diversity. Okay, so I'm going to get into some of the things that we're working on, and start with kind of a primer of salmon diversity. So when we think about diversity, probably the level that we most often immediately think about is species level diversity. And I've already shown you the 5 species of Pacific salmon plus our good friend the steelhead. But this is only a tiny sliver of the diversity. If you peel back the layers, there's much more diversity there within each of these species. So the first level is this geographic diversity. So for example, if you peel back the onion of diversity in Chinook salmon and you look across the range of Chinook salmon, well, notice there's genetically divergent or genetically distinct spawning populations that are separated geographically by watersheds. So each of these watersheds will have its own genetic grouping of salmon, and they're genetically distinct from other watersheds because of natal homing, right? Because they basically go back to the stream they were born to spawn, and that creates this population genetic structure within the range. Okay, then within each of these watershed genetic geographic groups, we can have migration diversity. So adults enter the rivers at different times. So some enter in winter, some enter in spring, some enter in fall, and some enter in late fall. And so each of those groups of fish is genetically distinct from others. And they lay eggs at slightly different times, and their juveniles migrate back to the ocean at slightly different times. All right, so that's another level. Then within each of these run types, we have age classes. We have some individuals maturing at age 2, age 3, age 4, age 5, or age 6. And so those are different groupings as well because, as I mentioned before, salmon reproduce once and die, right? So they don't reproduce multiple times. So these are not overlapping. They are discretely different maturation ages. Within each of these maturation ages, we have males and females. They show sexual dimorphism, right? So they show differences in their physical, behavioral, and physiological characteristics based on whether they're male or female. And then even within the sexes, we'll have different forms, like we'll have different reproductive types of males. And then all the way down to the individual genotype level. Of course, every individual is different from every other individual. And so we have this sort of hierarchy of diversity within Pacific salmon. Within any species of Pacific salmon, we have this hierarchy of diversity. For the purpose of this talk, I'm going to sort of categorize these groups into sort of 3 levels of diversity. So we have geographic diversity. I'm going to call sex, age structure, and migration. I'm going to call that discrete, ecological, and life history diversity. By discrete, I mean that you could assign a salmon individual to any one of these discrete categories, be it winter run, spring run, fall run, or late fall run. So those are discretely different categories. And then finally, what I call continuous adaptive diversity. So this is the continuous variation in traits that that you can't assign an individual discretely to one group or another, but they show continuous variation in those traits. And I'm going to start by describing a little bit of what we know about each of these levels of diversity. So the first is geographic diversity. So as I mentioned before, natal homing leads to reproductive isolation among these different spawning rivers. And so therefore, different rivers are genetically distinguishable from one another, and geneticists have been able to differentiate these rivers using neutral genetic markers for a long time. So some of these marker types are called microsatellites or single nucleotide polymorphisms, which we call SNPs. So there's a long history of people trying to understand what this population structure looks like across the landscape. And just one example I'll give you here is the population structure of Chinook salmon in the Yukon River drainage in Alaska and Canada. So you can see the lower Yukon, upper US Yukon, each of these groups, the Tanana, each of these groups is a genetically distinguishable group of fish that you could assign a fish. If you catch a fish down here on the lower Yukon River and you genotype that fish at these SNP markers, you can assign it to one of these spawning groups, okay? And the tools have been available to do this since the 1990s, so a lot of work has been done on this, and we know a lot about population genetic structure across all species of salmon and steelhead. One important thing is that because we have such a long history of understanding this level of diversity, it is really the historical backbone of genomic conservation as written into the U.S. Endangered Species Act. So these different genetic groupings can be considered what are called evolutionarily significant units, or ESUs, and under the Endangered Species Act, they can be protected individually. They can be given protection. So they can be given threatened status or endangered status, which allows conservationists to protect certain spawning rivers if those spawning rivers begin to experience declines. Okay, let's move to the next level, which I call ecological and life history diversity. So this is widespread variation in discrete traits such as migratory behavior, maturation age and size, and reproductive characteristics. And this is really where genomics has advanced over the last decade. So recent genomic advances have identified the sources of many of these discrete life history variants. So some of the really exciting papers have come out recently in this area. And I say that this has given rise to the supergene. So the supergene is this idea that there are genes of large effect that can determine these major life history variants. For example, a gene of large effect that will make the difference between an individual being a fall run or a spring run salmon. And importantly, they control these major traits, but they generally do not confer reproductive isolation. And so they're a bit of a problem for our protection paradigm under the Endangered Species Act through these ESUs, because these ESUs are predicated on the idea of reproductive isolation. So we have these major differences between, for example, here showing an anadromous or migratory steelhead versus a freshwater resident rainbow trout. This fish migrated to the ocean, spent several years growing in the ocean and returned to freshwater. This fish never left its freshwater stream, stayed there the whole time. Anadromous steelhead return, they encounter resident rainbow trout. They look super different, they act super different, they can spawn together. By all practical sort of purposes, they're very different fish, but they may only differ at one genetic region. And that genetic region is called this OMI5 supergene, and that's been recently identified by some of the collaborators that we have at the National Marine Fisheries Service. And they've identified that there are these two variants, resident and an anadromous variant of this gene. And depending on which variations of this gene these individuals inherit from their parents, goes a long way to explaining whether they migrate or they remain resident. Other exciting work that's been done recently is on run timing. So there are spring run Chinook salmon and fall run Chinook salmon. They enter rivers at different times. So spring run enter earlier in the season, fall run enter much later. And recently it's been uncovered that they differ at this OTS28 genomic region, and that has to do with the timing of maturity relative to freshwater entry. And then finally, some work, recent work's been done looking at maturation. And in particular, male maturation in Chinook salmon is driven largely by variation on the Y chromosome. And that will determine whether individuals mature young at ages 2 or 3 or mature late as old as age 5 or age 6. So these relatively simple and identifiable genomic regions underlie some of this really basic and pretty substantial behavioral variation, morphological variation, and life history variation. And then finally, sort of the most fine scale of adaptive diversity we have is this continuous adaptive diversity. So continuous variation in adaptive traits that is partitioned within and among populations. So many physiological and behavioral traits belong sort of in this category. Many of these traits determine survival and reproductive success. Less work has been done in this area, but we think that many of these traits are probably determined by the interacting effects of many genes. So not as simple as the super gene idea that we have for these major life history variants. And really the next frontier in this area requires that we do some experimental studies to measure these traits. And I'm going to give you one example that we're working on now, and then combining that with whole genome sequencing to then identify the genetic basis of variation in those traits. So one example, and this is Paige Gardner's PhD dissertation project. So Paige is looking at thermal tolerance. So steelhead ability to tolerate warming temperatures. And what Paige did was tested a bunch of different fish to see how hot could they take it, right? So she basically put a bunch of fish into a fish hot tub and heated them up until they couldn't take it anymore. They didn't die, but they do kind of tip over and tell you by going kind of belly up, I can't take it anymore, take me out. And so Paige takes out the fish when they get to that point and then records the temperature. Okay, so that's what we're showing you here, which is called the critical thermal maximum. And so you can see these dots are kind of all over the place. So you think like, oh my gosh, these dots are all over the place, right? But that's variation. That's actually probably, that's a good thing for the fish in that there are some fish across these populations that actually can tolerate pretty warm temperatures. And so we're trying to identify the genetic basis of that ability for those fish to tolerate those warm temperatures to see, because like, of course, our streams are warming, right? So how long are they going to be able to survive in our warming streams? And what that gets to is this final point. So a lot of the variation of these kind of traits is going to underlie future adaptive potential to tolerate these rapid environmental changes that we're experiencing. Okay, so I mentioned that salmon are sort of not doing super hot. Many populations are declining. And we have in sort of salmon speak the 5 H's. So people will talk about the 5 H's. So I'll tell you about the 5 H's. So these are the things that generally are causing salmon populations to decline. Hydro, so this is dams, water use, habitat, loss of habitat, harvest, so overharvest, removing too many fish from the population, hatcheries, which are often a direct result of dams and habitat loss. And then because climate change doesn't start with an H, we just called it heat, but you can think of heat as sort of our way of making climate change into an H, okay? And most of the time when we think about these or when people talk about these, the 5 H's, the thinking is about the impact of the 5 H's on population size or on abundance. But importantly, they also cause important effects to all these different levels of diversity that we talked about. So for example, when you build dams, it can cause the loss of anadromy in steelhead or the loss of early run timing in Chinook salmon. We talk about habitat loss. We can lose entire unique populations. Or we can lose freshwater rearing types, or types that spend a lot of their life history rearing in freshwater before they migrate to the ocean. When we harvest, we can remove the late maturation individuals because they're more vulnerable to harvest for a longer part of their adult lifespan, and so forth. So all these human impacts are causing not just declines in abundance, but also declines in diversity. And those declines in diversity are sort of happening at all these levels. So loss of geographic diversity across the landscape. So this paper goes back to 2007. So probably, things are probably somewhat worse now. But at least at this time, about 1/3 of historical populations had been extirpated. Interior spawning populations are in particular highly vulnerable. So interior spawning populations, for example, in the Columbia River Basin and in the San Joaquin River Basin of California have experienced a lot of extinctions. And increasingly, the southernmost populations, or those populations at the southernmost end of the distribution, are increasingly threatened. We've seen a loss of ecological and life history diversity, so decline of early run timing. In particular, spring run is highly threatened. We've seen declines in late maturation that have led to reduced age structure. So salmon are returning to spawn much younger now than they did historically. So those age 5-year-olds and age 6-year-olds, you'd have a hard time finding them in wild populations today. And then decline of prolonged freshwater rearing. So it's very hard now to find stream type juveniles because stream conditions are not able to sustain freshwater growth, and so they're migrating to the ocean at a younger age. And then, of course, that final level, loss of adaptive diversity within populations. We have hatcheries, homogenizing genetic variation. I'll talk a little bit about how that works in a second. And all of those things reduce future evolutionary potential. So why does this diversity matter, right? Why are we even concerned about it in the first place? Well, you may be concerned about it because diversity is the spice of life, but you also, if you're more into thinking about the value, like, well, what is the ecological value, or what is the value for people, right? Diversity has identifiable value. And so the paradigm that we think about when we think about this diversity is portfolio effects. And the portfolio, a salmon portfolio, is no different than a financial portfolio. So when you think about your financial portfolio, right, you think, okay, well, this portfolio only stocks, not diversified. What happens if the market crashes? Like, you lose all your money, right? Over here, you have a slightly more diversified portfolio. So now, maybe not quite so bad, but still vulnerable to changes in the market. But what you really want is this, right? A very diverse portfolio that includes many different stocks, many different bonds. And this diversified portfolio is going to be more stable and more resilient to perturbations, okay? So your financial advisor, if they're doing their job, is probably advising you to be over here, not over here. And the same thing is true conceptually when we think about salmon. So we can have one species and one run type of salmon, and that one population of totally homogeneous fish is going to be very vulnerable to perturbations or changes in the environment. When we have a highly diverse portfolio of salmon species and life histories within those species and genetic variation within those populations, now fluctuations in the environment are buffered by all that diversity. So if the environment gets warm, one form might decrease, but another form might like it warm and might do better. If the ocean gets hot, it might favor one form while another one does worse. And so this sort of portfolio effects idea underlies a lot of how we think about the value of salmon diversity. Okay, so now I'm going to sort of go through these different levels of diversity, now thinking about it through the lens of the ecological value that these levels of diversity confer, right? So here's this level, geographic diversity. So again, we're talking about differences in spawning rivers and regions. So this is really our classic example of salmon portfolio effects, where different spawning rivers contribute to that portfolio of different population types. And this is known to now maintain a stable supply of fish and nutrients for ecosystems and fisheries. So all those things I talked about before when it comes to marine predators and terrestrial predators and fertilization effects and salmon in trees and pollinators and plant interactions are all going to be stabilized when this salmon portfolio is more diverse and therefore more stabilized. So one example of this is actually from here, Central Valley, California Central Valley. And these portfolio effects were compared by Carlson and Satterwhite in the Sacramento Basin here and in the San Joaquin Basin here. So you can see in the Sacramento Basin, we have these populations are fluctuating, but they're fluctuating asynchronously, right? When some are up, others are down, but when then those go down, other ones go up. And the result is this relatively stable supply of fish, this blue line, maintaining those ecosystem services. In contrast, in the San Joaquin Basin, we get these boom-bust cycles, right? So we get periods of high abundance followed by crashes. And one difference between the Sacramento and the San Joaquin Basin is the hatchery influence in the San Joaquin Basin. It has an extremely large hatchery influence. And so therefore, high degree of synchronization, okay? So these boom-bust cycles are sort of eroding this portfolio effect. And things like dams, habitat loss, these lead to population extirpations, and things like hatcheries are known to homogenize populations by increasing rates of straying. So why do hatcheries do this? Because individuals that would normally go back to their natal stream to spawn, oftentimes if they're from a hatchery, they don't really know where to go, right? They're not sure what river to go to. So they'll make mistakes. And if they make more mistakes and they spawn across a whole variety of different places where they really shouldn't be spawning, you end up, instead of with a bunch of different populations that are all fluctuating asynchronously, with essentially one population that now fluctuates synchronously. Okay, let's move on to life history diversity and why that might matter. So one reason why the life history diversity matters is because many of these discrete variants have some unique value. And one example of that is spring run Chinook salmon. So spring run have particularly high fat content, high nutrient content that derives from the fact that they enter rivers when they're premature. So they haven't invested a lot of their bodily resources into reproduction yet. So they still have a lot of the fat that they're going to burn along their spawning migration, right? So they're entering the river at an earlier stage in the maturation process. And that leads them to have very high nutritional value at a very important time when people and ecosystems are reliant on that value, right? So they enter rivers right after winter, and they provide this sort of unique value in terms of their contributions to the ecosystem and to the fisheries by entering the rivers at that time. But we also have these portfolio effects. And for example, when we have individuals maturing at different ages, we can think of that as a temporal portfolio effect. So this is now spreading risk instead of across space, which is what the spatial portfolio effect I talked about before. This is a temporal portfolio effect. So it's spreading risk through time, right? So it means that different individuals from populations are going to be entering rivers in different years. So if there's a particularly bad year, it's not going to wipe out an entire cohort of fish if you have that age structure present in that population. If all the fish spawn the same year and that's a particularly bad year, that's going to cause a population crash. If maturation is spread across a bunch of different age classes, it's going to buffer that population from those fluctuations. And this is an example of a paper that my former postdoc, Paul Carvalho, did, where he specifically looked at the contribution of age structure to this temporal portfolio effect and found kind of that the prediction held pretty much true. That if you have only one age class, as shown here, you have dramatic fluctuations in the population. If you have a multitude of age classes, like here or here, you basically have a much more stable population. How does this occur? Well, how can you this sort of gets to the restoration bit, which is like, well, how do we restore population structure? Well, one way is to reduce mortality. So if we do reduce fisheries mortality, we can restore age structure. But if you don't reduce fishing mortality, one thing we observe is fish will just evolve away from the fishery, right? And if fish evolve away from the fishery, then we can also have a situation like this. And this is really kind of the area that my lab has been focusing a lot of our work over the last few years on, this area of the ecological effects of this life history diversity. And we've looked at this in a variety of different places. So we looked at this in Alaska in terms of the effects of fishing and climate on body size and the effects that that has on ecosystems and fisheries in Alaska. We've looked at this in California steelhead populations and their resilience to drought. Here's Paul's paper here, which I just talked about. And then we've looked at this in terms of the anadromous and resident steelhead case example that I talked about before. So this is kind of a big area of our research focus right now. And then kind of like the next frontier of thinking about things moving forward is really this continuous adaptive diversity. So again, now we're talking about this continuous adaptive genetic variation, which is the raw material for adaptive evolution. So if you remember back to maybe if you took like evolution class in college, right? Think about natural selection. Well, this genetic variation is the raw material that selection needs to act. And so it's critical for population persistence because populations need to be able to evolve to persist in a changing environment. So here we have a situation, this is called sort of a theoretical situation called evolutionary rescue. So what this shows is an environmental change here that causes our population size to decline rapidly. But if that environmental change is in a certain direction for which there is adaptive genetic variation shown down here, right? So this is selection acting to favor that trait. So let's say this is a stream warms up, right? One of Page's study streams warms up, but we have these individuals in that population that are able to tolerate warm temperatures. They're going to be favored, right? They're going to reproduce, and the next generation of fish is going to be more heat tolerant. Okay, so as the population declines, the trait value is going to move in this direction towards the new environment. As this trait value moves towards the new environment, it's going to cause this population ultimately to start to recover as the population becomes more suited to that new environment, right? As selection acts, and eventually you're going to get a population that has a high degree of adaptation to the new environment, and now the population size can recover. And that's a dynamic evolutionary process that we want to maintain the genetic variation in wild populations so that that natural process can happen as environments change. Okay. So in terms of our genes to ecosystem now restoration approach, right? I'm going to try to put this all together and try to come up with some rules for restoration where we can use this information about genetics, use this information about ecology, and come up with some take-home points for how do we do restoration or how do we transform salmon restoration in moving forward in a rapidly changing environment. So I want to start with something that's not so new, which is Aldo Leopold, right? And Aldo Leopold said the first rule of intelligent tinkering is to keep every cog and wheel, right? So don't throw away any of the parts. So that is not a new insight, but I think it's still a very important insight that we have to keep in mind all the time. In terms of thinking about restoration targets, I think what all this means is that we need to think about restoring diversity, not just fish numbers. So it's not just the number of fish that are in your spawning river or not just the number of fish that you can catch in the ocean, but it has a lot to do with the diversity of those fish. And the diversity of those fish is going to go a long way to determining how resilient those populations are to environmental change moving forward. In order to get there, I think we need to develop genetic management strategies. So we need to be able to identify adaptive alleles, and we need to be able to move them into at-risk populations, right? So we need to actually be able to kind of be proactive and think a little bit forward. How are environments changing? What kind of traits will populations need in the future? And can we move those adaptive alleles into those populations proactively so natural selection has the raw material it needs to act? And all this is towards restoring dynamically evolving populations. So say populations cannot exist or persist as postage stamps on the landscape, isolated from all other populations. They have to be dynamically evolving. I would say it's probably a pretty good rule of thumb to say we should go for habitat over hatcheries. So habitat restoration is a better bet than restoration through hatcheries. Hatcheries can be a short-term strategy at best, but hatcheries are not a long-term solution. And we need to, in the case of reintroduction, so now we have cases where populations have been completely lost from many spawning rivers. So what are we going to do there? So when we go to do reintroductions, we need to keep diversity in mind in those introductions. So we need to reintroduce diverse founder populations with high adaptive potential. We don't want to introduce all fish that are genetically identical to one another. We want to introduce genetic variation that future selection can act on. And I think when we set our restoration targets, we need to set our restoration targets with all these levels of diversity in mind. And just 2 projects we're working on now to mention. So you may have heard of the Klamath River Restoration Project. So the largest dam removal project in U.S. history, sort of underway, dams removed now. Now the sort of additional steps of restoration can start to take place. And one of the primary goals of that effort is to restore spring run Chinook salmon. So that project is very specifically taking into account this life history variation and the particular ecological and fishery value that life history variation brings. And then down here, Halama Creek steelhead reintroduction. So this is a project that we're undertaking. I should mention this Klamath River restoration. We have NOAA fisheries partners working on that, and we have Yurok tribal fisheries partners working on that project with us as well. Here we have the Halama Creek Steelhead Restoration Project, and this is a partnership with the Nature Conservancy. This is a stream in Southern California that once had steelhead, endangered form of steelhead, Southern California steelhead, that went extinct, and we're in the process of developing a plan to reintroduce steelhead to that stream. So we're going through that process now of trying to restore habitat and prepare it for reintroduction of steelhead. So super exciting projects where we can put these sort of lessons learned, all these things that our science has been pointing us to, into effect through these actual on-the-ground restoration projects. Okay, so progress and future needs. So I want to say a few words about sort of where we are now and sort of where we need to go generally speaking. So if we sort of think about these different levels of variation that I talked about before, right? So we have geographic, yikes, I went forward too fast. If we have geographic diversity across the landscape, we have discrete ecological and life history diversity, and we have continuous adaptive diversity. And we think about sort of how we have applied genomic tools to address these levels of diversity, our understanding of their ecological effects, the conservation frameworks that we have to implement this understanding, and then applications to on-the-ground restoration. I'm going to take like a red light, yellow light, green light approach to sort of saying like, how are we doing? Like, how much do we know? How much of this can be implemented? And as I said earlier, when it comes to this geographic diversity across the landscape, we've known about this for a long time. So we have very well-developed paradigms for thinking about this. The genomic tools are widely available and have been widely applied. We understand a lot about the ecological effects through our understanding of portfolio effects. I give this one sort of a green-yellow, though, because although we understand these effects and how they might happen, there's only a few places where we've really implemented that understanding. So we really need to apply that much more broadly to a bunch of different locations and a bunch of different species. We have well-developed conservation frameworks through the application of evolutionary significant units as applied in the Endangered Species Act. So we have a very solid conservation framework for thinking, and we have started to take approaches to restoration. Okay, so now as we move through these different levels, things are going to get a little more shaky, right? Our understanding is getting a little more shaky because the science is newer, right? As I mentioned, the science of understanding the variation, the genomic variation underlying these discrete ecological and life history diversity is much newer, right? All these insights come from about the past 5 to 10 years. So we have a lot more to learn there, a lot more traits to look into. Our understanding of the ecological effects is coming along. This is where my lab is contributing a lot of the understanding that we currently have. But again, there's a lot more we have to learn. Our conservation framework is challenged by these kinds of traits because, as I mentioned before, they have a big impact on phenotypes or the traits that we observe, the morphological traits, the behavioral traits, the physiological traits, but they don't fit neatly into the conservation paradigm that we currently have. So we're going to need to adjust our conservation paradigm to kind of figure out how to fit these traits into kind of the conservation framework that we have. A good example of that is the controversy over whether we should list spring-run Chinook salmon under the Endangered Species Act, and there's a lot of controversy over that. And then finally, restoration applications. And we're sort of getting there, right? As I mentioned, we are focusing in some cases on restoring specific life history variants. So we're making some progress in that area. And here's where things need a lot of progress, right? Continuous adaptive diversity within and among populations. So I sort of give us on application of genomic tools like sort of a yellow-red. I give us the yellow because we kind of know what we have to do, right? We kind of know the tools and techniques we're going to have to apply. I give us a red because we have a lot of work to do to actually apply them. And so, like, for example, Paige's project is going to be super novel and super exciting because it'll be our first insight into the adaptive genetic basis of thermal tolerance in steelhead. So we'll learn something brand new about how steelhead may or may not be able to evolve to warming stream temperatures. We have much less of an understanding of ecological effects. Our understanding is largely theoretical, but we haven't done a lot of the experiments we need to do to test them. Our conservation frameworks are not really up to the task of taking contemporary evolutionary change into account and thinking about how to move adaptive alleles around the landscape. Right now, in fact, you largely can't do that, right? You largely can't move salmon from one population to another. It's something that we need to think deeply about. Is that the right way to go forward? And in terms of restoration applications, sort of same situation. We need to do some more thinking about how to integrate this level into restoration. There's so much more I could have said about other exciting projects going on in my lab. All of my PhD students and postdocs are doing absolutely amazing cutting-edge research. They're working on a whole variety of things from wildfire effects to the effects of individual genes on entire ecosystems to major problems we're facing like thymine deficiency, thermal tolerance, which I talked a lot about, steelhead reintroduction and how we're going to do that, and even salamanders. And you can ask me about salamanders later. And if you've been following the news, I just want to say it's not people over fish, it's people and fish. And with that, thank you for your attention. I'd like to thank all the members of my lab, the Fisheries Collaborative Program, which is the program Brian mentioned that I'm the director of, and the administrative assistants of that program, Sarah Nestle Bauer and Jaylene Alonso. UCSC Center for Coastal Climate Resilience has supported a lot of our work. We do a lot of this work in partnership with NOAA, National Marine Fisheries Service, through the Cooperative Institute, and in collaboration with the Southwest Fisheries Science Center. I want to give a particular shout out to Carlos Garza and Steve Munch, are two of my very close collaborators at NOAA. Our work in Halama Creek with the Nature Conservancy, and our work on the Klamath River with the Yurok Tribal Fisheries Department. And I'd be happy to take questions, right? Okay, thank you.
Grab a seat.
All right.
All right, so that was amazing. I know so much more about this than I did 45 minutes ago. So we've got lots of time for questions, both from the audience in person and from the internet. So questions for Eric, and just wait for the microphone before you ask your questions so that those listening online can hear it. Yeah, so right in the front here, and then we'll go to the back next.
So I have 3 million questions, but I'll... So just some molecular questions. how much of the genetic diversity is due to epigenetic changes rather than nucleotide changes? And then the second part of that is whether the populations are at all amenable to biobanking and other molecular manipulations.
Yeah. Okay, epigenetics. So that's probably like the next level, right? So we had level 1, level 2, level 3. You asked about level 4. So our red light, green light went like sort of green, yellow, red, like epigenetics, red. Like we have a lot to learn, right? We have a lot to learn about what epigenetic control, so thermal tolerance may have an epigenetic component. And we largely don't know what that component might be. So there's a lot of work we have to do there. In terms of biobanking, so I think what you're sort of referring to is like, can we preserve some of this? Can we sort of archive some of this variation so that sort of if these populations are lost, the underlying genetic material is not lost? And the answer is yes. Like that is definitely something we should be doing and are doing. I personally think we should be taking an approach where we keep adaptive alleles alive in living fish so if we know that, let's say we know conditions are going to become too warm in a certain stream for the fish in that stream to tolerate it. Yet those fish may very likely have the alleles for high thermal tolerance, but that stream is just going to get too hot. But the probability is that streams further north are also going to need that adaptive genetic variation to underlie their future adaptation. So one strategy we could think about taking is moving fish from endangered warm southern streams further north to try to introduce those alleles into other populations where they can sort of accelerate evolutionary potential moving forward in streams that may not exceed thermal tolerance limits, but nonetheless are going to need to go through some adaptive evolution to persist.
All right, so I've got a question all the way in the back corner there.
My question is about reintroduction of the favorable species in certain areas. First question is, how do you manage any risks from bears or any other species that might be competing for a certain part of the creek, for example? And second question, how exactly do you reintroduce those species? Do you put the fish eggs there or like full-grown fish or,
Great questions. Okay, so how do you manage risks associated with natural predators and other things that might want to eat the fish that you're trying to reintroduce? Largely we have to have strategies that are resilient to those things. I mean, that's the way the ecosystems work. And ultimately, the value that salmon provide for predators is part of the ecological value of having salmon. So we don't want to prevent bears from eating salmon. We want to have a resilient salmon population that can feed bears, okay? So getting a new population started, though, where you only have a few individuals can be tricky, right? It's like, how do you get over that initial sort of hurdle of getting that population starting to grow? That is a challenge. So right now, that's what we're thinking about. For example, for Halama Creek is like, how many fish do we need to introduce? At what life stage do we need to reintroduce them? Where should we put them? Do we introduce spawning adults? Do we introduce juveniles? These are the things that we're trying to think through right now. One of the things I can say is, sometimes this is all taken out of your control because the source of those fish may be not entirely up to you. So one of the recent things that happened is the Palisades fire burned one of the last remaining strongholds for Southern California steelhead in Topanga Creek. And so there were all of a sudden the need to move all these fish out of that watershed and put them somewhere else to save those fish. And so we've been sort of going through the process of evaluating whether Halama Creek might be a suitable location to translocate those fish. And we essentially have no control over that process. It was a natural disaster that led to this acute restoration. In one case, an opportunity resulting from sort of a major threat. So how to best do those things, we're thinking about them. Sometimes we don't have total control over how we will do them. Yeah.
Just a follow-up from that, at what point in the life cycle does the salmon sort of imprint as to where it has to go back to? Is it when they hatch or during that maturation period?
Yeah, it's during that juvenile period, right, when they're living in the stream. And in fact, Morgan Abbott, one of my PhD students, is working on that specific question right now, looking at gene expression in the olfactory system that's associated with natal imprinting and natal homing. And so it's through that juvenile development period, they sort of, the sort of olfactory cues of their natal stream sort of create a signature in their brain that they can detect when they return, so they can identify what stream they need to spawn in when they return as adults. And one of the things that Morgan's investigating is whether wildfire, or the effects of wildfire and the resulting sort of flow of debris and other olfactory, potentially contaminants into rivers might interfere with that imprinting process. Right. So open question. We don't know the extent to which that might be happening, but it's an important thing to figure out because wildfires are becoming increasingly common. And as they become increasingly common, we might end up with steelhead that can't find their way back home.
Matt, do we have any questions from the internet?
Yeah, we have a number of questions. A number of people asked a question about hatcheries. So I want to ask you if you could talk a little bit more about the threats that salmon hatcheries pose to salmon diversity. And is there anything that hatcheries can do to increase diversity and life history variation?
Yeah. So there's a couple of things that hatcheries do. So hatcheries are most efficient if they sort of produce a uniform product, right? And so a lot of the hatchery operations sort of are very sort of consistent, and because they're consistent, they are inadvertently imposing selection on populations essentially to be adapted to the hatchery, right? And so being adapted to the hatchery means that they're homogeneous in many ways, and they may not be adapted for sort of spawning in a wild situation. The other thing is that hatchery fish often don't imprint as precisely on their natal stream, and so they stray at higher rates, and that homogenizes populations. Okay, so what can be done about these things? First, in order to sort of prevent hatchery adaptation, we can continuously introduce wild fish back into the hatchery population. So that can sort of continuously infuse wild genes, wild alleles back into hatchery stocks. We can also make sure that hatchery fish do imprint on their natal streams. So strategies that sort of take salmon directly to the ocean, for example, completely avoid that imprinting process. And then you do end up with lost salmon, right? They can't find their way home. So sort of one of the things hatcheries can do is to make sure that juveniles coming from those hatcheries do have an opportunity to imprint on their natal streams.
Do we have more questions from this side? Yep, great, over there, thank you.
Hi, thank you. I'm so curious about the difference between the sort of reintroducing adaptive alleles, where you're moving fish from one place to another for population like robustness, versus a lost fish phenomenon, like how you're moving fish from one place to another and you're expecting the populations to mingle and blend, or you worry about supplanting that as well, but how do you distinguish what is going to be a positive effect versus the lost fish effect?
Right. So these are like the opposite sides of the same coin, right? So basically, if gene flow homogenizes, but gene flow also sort of potentially spreads adaptive alleles. So what that means is that we sort of need to think carefully through how we're doing this, right? Because the inadvertent sort of bad side effect, and this is why this is like not what we currently do, right? And that's why probably if like I have some colleagues like watching this talk, they're like sort of like feeling a little uncomfortable about moving fish around, because it definitely does kind of, it means that there potentially would be higher straying rates. However, sort of this is my argument, is that for some populations, we're in a situation where if we do nothing, they're going to go extinct, right? And so if we're in a situation where if we do nothing, they're going to go extinct. And not only are we going to lose a population in that creek, but we're going to lose the adaptations that that population has. I think we should do something to save those, if nothing else, to save that adaptive variation so that it can benefit the future adaptation of other populations.
The potential side effects of homogeneity is worth the risk when the alternative is losing.
That's what I would argue, and people will argue against that.
Matt, we got any other questions from the cloud?
Yeah, there's a number of other questions. One, we appreciate all the great work that you're doing and your lab is doing, but what can citizens do to help support salmon recovery?
Yeah, good questions. As I sort of alluded to at the end, so these restoration projects, they are big projects that require lots of partners, right? So they don't happen because sort of an academic institution decides we want to do a restoration project. They happen because we talk to others who are on the ground doing restoration work, like the Nature Conservancy, right? Like NOAA Fisheries, like a lot of local watershed groups that have a vested interest in saving their salmon. And we have lots of those kind of organizations in the Monterey Bay Area, and in the San Francisco Bay Area. So getting involved with those local organizations is really a good way to sort of make an impact, because really these restoration projects happen through partnerships. And it takes all these partners working together, sort of doing what they do best to make these things happen. And the thing that we do best is sort of we do the science, right? And we kind of try to help understand how we should do these projects. But when it comes to doing these on-the-ground projects, we need the resource conservation districts, and we need the NGOs, and we need our federal partners, and we need our state partners, and we need our tribal partners. It takes everyone working together to pull these things off.
Okay, we've got time for one more question. Is there anyone who hasn't asked a question yet who wants to ask one? Yeah. Someone, you already handed out the microphone? Yeah, go ahead.
Yeah, hello. First off, pleasure to meet you, and thank you for the presentation. The question that I have is in terms of restoration. Like, what methodologies are you using to monitor the success rate of your restoration efforts, and how are you quantifying whether something is successful or not successful?
Yeah, so I didn't plant that question, but I appreciate it, because another sort of slide I cut, another sort of recent advance in genomics is really the ability to use genotypes to identify down to the level of individuals. And so one of the techniques we can use is genetic mark recapture. So we can basically take a tiny, tiny piece of tissue from a fish that we catch in our fish surveys, and that essentially serves as an individual tag. And we now could re-identify that fish. If we go back and resample that location, we'll know that that's the same fish we caught before. So that's much less invasive than traditional tagging approaches, which would involve doing something like implanting like a pit tag, like a digital tag into a fish that then you have a reader and you can read the fish's number. Like your pets have like the same thing, right? But the genetic tags are much less invasive, especially for small fish, so have much less harmful side effects. What that allows us to do is use mark recapture tools to estimate population size. And so we can understand whether the population is sort of declining or sort of holding steady, or if we're getting population growth.
All right, so thank you for all the questions. I'm going to hand over in a moment to our host, George Kraw, to wrap things up. Before we do, I just want to thank you again for coming. I just want you to please thank our events and our AV staff for making everything run so flawlessly. Amazing questions. I always love getting questions from people in the Silicon Valley and the Bay Area. They're like just a cut above. So over to you, George.
Yeah, well, thank you very much for that fascinating conversation tonight. And thank you all for coming. Please join us on March 5th for our next lecture, which will be on utilizing and unlocking the potential of AI for autonomous systems. Good night, everyone.